Cell biology. The cell, from Latin cella, meaning small room, is the basic structural, functional, and biological unit of all known organisms. A cell is the smallest unit of life. Cells are often called the building blocks of life. The study of cells is called cell biology, cellular biology, or cytology. Cells consist of cytoplasm enclosed within a membrane, which contains many biomolecules such as proteins and nucleic acids. Most plant and animal cells are only visible under a microscope, with dimensions between 1 and 100 micrometers. Organisms can be classified as unicellular, consisting of a single cell such as bacteria, or multicellular, including plants and animals. Most unicellular organisms are classed as microorganisms. The number of cells in plants and animals varies from species to species. It has been estimated that humans contain somewhere around 40 trillion, 4 times 10^13, cells. The human brain accounts for around 80 billion of these cells. Cells were discovered by Robert Hooke in 1665, who named them for their resemblance to cells inhabited by Christian monks in a monastery. Cell theory, first developed in 1839 by Matthias Jacob Schleiden and Theodor Schwann, states that all organisms are composed of one or more cells, that cells are the fundamental unit of structure and function in all living organisms, and that all cells come from pre-existing cells. Cells emerged on Earth at least 3.5 billion years ago. Cell types. Cells are of two types, eukaryotic, which contain a nucleus, and prokaryotic, which do not. Prokaryotes are single-celled organisms, while eukaryotes can be either single-celled or multicellular. Prokaryotic cells. Prokaryotes include bacteria and archaea, two of the three domains of life. Prokaryotic cells were the first form of life on Earth, characterized by having vital biological processes including cell signaling. They are simpler and smaller than eukaryotic cells, and lack a nucleus, and other membrane-bound organelles. The DNA of a prokaryotic cell consists of a single circular chromosome that is in direct contact with the cytoplasm. The nuclear region in the cytoplasm is called the nucleoid. Most prokaryotes are the smallest of all organisms, ranging from 0.5 to 2.0 micrometers in diameter. A prokaryotic cell has three regions. Eukaryotic cells. Plants, animals, fungi, slime molds, protozoa, and algae are all eukaryotic. These cells are about 15 times wider than a typical prokaryote and can be as much as 1,000 times greater in volume. The main distinguishing feature of eukaryotes as compared to prokaryotes is compartmentalization, the presence of membrane-bound organelles, compartments, in which specific activities take place. Most important among these is a cell nucleus, an organelle that houses the cell's DNA. This nucleus gives the eukaryote its name, which means true kernel, nucleus. Other differences include subcellular components. All cells, whether prokaryotic or eukaryotic, have a membrane that envelops the cell, regulates what moves in and out, selectively permeable, and maintains the electric potential of the cell. Inside the membrane, the cytoplasm takes up most of the cell's volume. All cells, except red blood cells which lack a cell nucleus and most organelles to accommodate maximum space for hemoglobin, possess DNA, the hereditary material of genes, and RNA, containing the information necessary to build various proteins such as enzymes, the cell's primary machinery. There are also other kinds of biomolecules in cells. This article lists these primary cellular components, then briefly describes their function. Membrane. The cell membrane, or plasma membrane, is a biological membrane that surrounds the cytoplasm of a cell. In animals, the plasma membrane is the outer boundary of the cell, while in plants and prokaryotes it is usually covered by a cell wall. This membrane serves to separate and protect a cell from its surrounding environment and is made mostly from a double layer of phospholipids, which are amphiphilic, partly hydrophobic and partly hydrophilic. Hence, the layer is called a phospholipid bilayer, or sometimes a fluid mosaic membrane. Embedded within this membrane is a macromolecular structure called the porosome, the universal secretory portal, and cells in a variety of protein molecules that act as channels and pumps that move different molecules into and out of the cell. The membrane is semi-permeable, and selectively permeable, in that it can either let a substance, molecule or ion, pass through freely, pass through to a limited extent, or not pass through at all. Cell surface membranes also contain receptor proteins that allow cells to detect external signaling molecules such as hormones. Cytoskeleton. The cytoskeleton acts to organize and maintain the cell shape, anchors organelles in place, helps during endocytosis, the uptake of external materials by a cell, and cytokinesis, the separation of daughter cells after cell division, and moves parts of the cell in processes of growth and mobility. The eukaryotic cytoskeleton is composed of microfilaments, intermediate filaments and microtubules. There are a great number of proteins associated with them, each controlling a cell's structure by directing, bundling, and aligning filaments. The prokaryotic cytoskeleton is less well studied but is involved in the maintenance of cell shape, polarity and cytokinesis. The subunit protein of microfilaments is a small, monomeric protein called actin. The subunit of microtubules is a dimeric molecule called tubulin. Intermediate filaments are heteropolymers whose subunits vary among the cell types in different tissues. But some of the subunit protein of intermediate filaments include vimentin, desmin, lamin, lamins A, B and C, keratin, multiple acidic and basic keratins, neurofilament proteins, NFL, NFM. Genetic material. Two different kinds of genetic material exist: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Cells use DNA for their long-term information storage. The biological information contained in an organism is encoded in its DNA sequence. RNA is used for information transport, for example, mRNA, and enzymatic functions, for example, ribosomal RNA. Transfer RNA, TRNA, molecules are used to add amino acids during protein translation. Prokaryotic genetic material is organized in a simple circular bacterial chromosome in the nucleoid region of the cytoplasm. Eukaryotic genetic material is divided into different, linear molecules called chromosomes inside a discrete nucleus, usually with additional genetic material in some organelles like mitochondria and chloroplasts. See endosymbiotic theory. A human cell has genetic material contained in the cell nucleus, the nuclear genome, and in the mitochondria, the mitochondrial genome. In humans, the nuclear genome is divided into 46 linear DNA molecules called chromosomes, including 22 homologous chromosome pairs and a pair of sex chromosomes. The mitochondrial genome is a circular DNA molecule distinct from the nuclear DNA. Although the mitochondrial DNA is very small compared to nuclear chromosomes, it codes for 13 proteins involved in mitochondrial energy production and specific tRNAs. Foreign genetic material, most commonly DNA, can also be artificially introduced into the cell by a process called transfection. This can be transient, if the DNA is not inserted into the cell's genome, or stable, if it is. Certain viruses also insert their genetic material into the genome. Organelles. Organelles are parts of the cell which are adapted and/or specialized for carrying out one or more vital functions, analogous to the organs of the human body, such as the heart, lung, and kidney, with each organ performing a different function. Both eukaryotic and prokaryotic cells have organelles, but prokaryotic organelles are generally simpler and are not membrane-bound. There are several types of organelles in the cell. Some, such as the nucleus and Golgi apparatus, are typically solitary, while others, such as mitochondria, chloroplasts, peroxisomes and lysosomes, can be numerous, hundreds to thousands. The cytosol is the gelatinous fluid that fills the cell and surrounds the organelles. Eukaryotic. Eukaryotic and prokaryotic. Structures outside the cell membrane. Many cells also have structures which exist wholly or partially outside the cell membrane. These structures are notable because they are not protected from the external environment by the semi-permeable cell membrane. In order to assemble these structures, their components must be carried across the cell membrane by export processes. Cell wall. Many types of prokaryotic and eukaryotic cells have a cell wall. The cell wall acts to protect the cell mechanically and chemically from its environment, and is an additional layer of protection to the cell membrane. Different types of cell have cell walls made up of different materials. Plant cell walls are primarily made up of cellulose, fungi cell walls are made up of chitin, and bacteria cell walls are made up of peptidoglycan. Prokaryotic. Capsule. A gelatinous capsule is present in some bacteria outside the cell membrane and cell wall. The capsule may be polysaccharide as in pneumococci, meningococci, or polypeptide as Bacillus anthracis, or hyaluronic acid as in streptococci. Capsules are not marked by normal staining protocols and can be detected by India ink or methyl blue, which allows for higher contrast between the cells for observation. 87. Flagella. Flagella are organelles for cellular mobility. The bacterial flagellum stretches from cytoplasm through the cell membranes and extrudes through the cell wall. They are long and thick thread-like appendages, protein in nature. A different type of flagellum is found in archaea and a different type is found in eukaryotes. Fimbriae. A fimbria, plural fimbriae, also known as a pilus, plural pili, is a short, thin, hair-like filament found on the surface of bacteria. Fimbriae are formed of a protein called pilin, antigenic, and are responsible for the attachment of bacteria to specific receptors on human cells, cell adhesion. There are special types of pili involved in bacterial conjugation. Cellular processes. Replication. Cell division involves a single cell, called the mother cell, dividing into two daughter cells. This leads to growth in multicellular organisms, the growth of tissue, and to procreation, vegetative reproduction, in unicellular organisms. Prokaryotic cells divide by binary fission, while eukaryotic cells usually undergo a process of nuclear division, called mitosis, followed by division of the cell, called cytokinesis. A diploid cell may also undergo meiosis to produce haploid cells, usually 4. Haploid cells serve as gametes in multicellular organisms, fusing to form new diploid cells. DNA replication, or the process of duplicating a cell's genome, always happens when a cell divides through mitosis or binary fission. This occurs during the S phase of the cell cycle. In meiosis, the DNA is replicated only once, while the cell divides twice. DNA replication only occurs before meiosis I. DNA replication does not occur when the cells divide the second time, in meiosis II. Replication, like all cellular activities, requires specialized proteins for carrying out the job. DNA repair. In general, cells of all organisms contain enzyme systems that scan their DNA for damages and carry out repair processes when damages are detected. Diverse repair processes have evolved in organisms ranging from bacteria to humans. The widespread prevalence of these repair processes indicates the importance of maintaining cellular DNA in an undamaged state in order to avoid cell death or errors of replication due to damages that could lead to mutation. E. coli bacteria are a well-studied example of a cellular organism with diverse well-defined DNA repair processes. These include 1. Nucleotide excision repair, 2. DNA mismatch repair, 3. Non-homologous end joining of double-strand breaks, 4. Recombinational repair, and 5. Light-dependent repair, photoreactivation. Growth and metabolism. Between successive cell divisions, cells grow through the functioning of cellular metabolism. Cell metabolism is the process by which individual cells process nutrient molecules. Metabolism has two distinct divisions: catabolism, in which the cell breaks down complex molecules to produce energy and reducing power, and anabolism, in which the cell uses energy and reducing power to construct complex molecules and perform other biological functions. Complex sugars consumed by the organism can be broken down into simpler sugar molecules called monosaccharides, such as glucose. Once inside the cell, glucose is broken down to make adenosine triphosphate, ATP, a molecule that possesses readily available energy, through two different pathways. Protein synthesis. Cells are capable of synthesizing new proteins, which are essential for the modulation and maintenance of cellular activities. This process involves the formation of new protein molecules from amino acid building blocks based on information encoded in DNA/RNA. Protein synthesis generally consists of two major steps: transcription and translation. Transcription is the process where genetic information in DNA is used to produce a complementary RNA strand. This RNA strand is then processed to give messenger RNA, mRNA, which is free to migrate through the cell. mRNA molecules bind to protein RNA complexes called ribosomes located in the cytosol, where they are translated into polypeptide sequences. The ribosome mediates the formation of a polypeptide sequence based on the mRNA sequence. The mRNA sequence directly relates to the polypeptide sequence by binding to transfer RNA, tRNA, adapter molecules in binding pockets within the ribosome. The new polypeptide then folds into a functional three-dimensional protein molecule. Motility. Unicellular organisms can move in order to find food or escape predators. Common mechanisms of motion include flagella and cilia. In multicellular organisms, cells can move during processes such as wound healing, the immune response, and cancer metastasis. For example, in wound healing in animals, white blood cells move to the wound site to kill the microorganisms that cause infection. Cell motility involves many receptors, cross-linking, bundling, binding, adhesion, motor and other proteins. The process is divided into 3 steps: protrusion of the leading edge of the cell, adhesion of the leading edge and adhesion at the cell body and rear, and cytoskeletal contraction to pull the cell forward. Each step is driven by physical forces generated by unique segments of the cytoskeleton. Multicellularity. Cell specialization. Multicellular organisms are organisms that consist of more than one cell, in contrast to single-celled organisms. In complex multicellular organisms, cells specialize into different cell types that are adapted to particular functions. In mammals, major cell types include skin cells, muscle cells, neurons, blood cells, fibroblasts, stem cells, and others. Cell types differ both in appearance and function, yet are genetically identical. Cells are able to be of the same genotype but of different cell type due to the differential expression of the genes they contain. Most distinct cell types arise from a single totipotent cell, called a zygote, that differentiates into hundreds of different cell types during the course of development. Differentiation of cells is driven by different environmental cues, such as cell-cell interaction, and intrinsic differences, such as those caused by the uneven distribution of molecules during division. Origin of multicellularity. Multicellularity has evolved independently at least 25 times, including in some prokaryotes, like cyanobacteria, myxobacteria, actinomycetes, magnetoglobus multicellularis or Methanosarcina. However, complex multicellular organisms evolved only in 6 eukaryotic groups: animals, fungi, brown algae, red algae, green algae, and plants. It evolved repeatedly for plants, chloroplastida, once or twice for animals, once for brown algae, and perhaps several times for fungi, slime molds, and red algae. Multicellularity may have evolved from colonies of interdependent organisms, from cellularization, or from organisms in symbiotic relationships. The first evidence of multicellularity is from cyanobacteria-like organisms that lived between 3 and 3.5 billion years ago. Other early fossils of multicellular organisms include the contested group Aniospiralis and the fossils of the black shales of the Paleoproterozoic Francivillian group fossil B formation in Gabon. The evolution of multicellularity from unicellular ancestors has been replicated in the laboratory, in evolution experiments using predation as the selective pressure. Origins. The origin of cells has to do with the origin of life, which began the history of life on Earth. Origin of the first cell. There are several theories about the origin of small molecules that led to life on the early Earth. They may have been carried to Earth on meteorites, see Murchison meteorite, created at deep-sea vents, or synthesized by lightning in a reducing atmosphere, see Miller-Urey experiment. There is little experimental data defining what the first self-replicating forms were. RNA is thought to be the earliest self-replicating molecule, as it is capable of both storing genetic information and catalyzing chemical reactions, see RNA world hypothesis, but some other entity with the potential to self-replicate could have preceded RNA, such as clay or peptide nucleic acid. Cells emerged at least 3.5 billion years ago. The current belief is that these cells were heterotrophs. The early cell membranes were probably more simple and permeable than modern ones, with only a single fatty acid chain per lipid. Lipids are known to spontaneously form bean-layered vesicles in water, and could have preceded RNA, but the first cell membranes could also have been produced by catalytic RNA, or even have required structural proteins before they could form. Origin of eukaryotic cells. The eukaryotic cell seems to have evolved from a symbiotic community of prokaryotic cells. DNA-bearing organelles like the mitochondria and the chloroplasts are descended from ancient symbiotic oxygen-breathing proteobacteria and cyanobacteria, respectively, which were endosymbiosed by an ancestral Archaean prokaryote. There is still considerable debate about whether organelles like the hydrogenosome predated the origin of mitochondria, or vice versa. See the hydrogen hypothesis for the origin of eukaryotic cells. History of research. Eukaryotic cells. History of research. Eukaryotic cells. History of research.